Good morning or afternoon, evening, everyone. I'm calling in from Singapore, so it's late for me, but good to see some folks on this call. For anyone that's new or attending this call for the first time, so we hold this industry-wide call every month for companies that are all along the cultivated meat value chain. And the purpose of these calls is really to bring in different solutions providers into the cultivated meat ecosystem, with the end goal of hopefully establishing some new collaborations between companies that are already in the cultivated meat sector and those that are developing technologies or providing services that can accelerate the industry. So if your group is interested in presenting on a future call, or maybe you're working with another company that may benefit from being on these calls, please feel free to reach out to me directly. I'm happy to chat with them and share some details. So just a few housekeeping items on our end. We are currently conducting this survey on cell lines. You may have seen some emails already from Renee. We're trying to get responses collected by the end of the month, although we might extend that deadline. But in summary, we're teaming up with some researchers at A-Star in Singapore to survey the industry on questions related to cell line development needs and trends and different regulatory issues. And having this data firsthand from the industry is going to be really impactful for a lot of different stakeholders in the industry. So I would really encourage you to take the time to complete it or forward it to the appropriate person on your team if you're doing anything that's related to cell lines. The survey takes about 30, maybe 40 minutes to complete. The results are going to be aggregated. Your responses are confidential or de-identified. They're only visible to me and Dean, who's my colleague that's helping me run this project. And I'll drop that link in the chat. And I want to thank we have already 18 companies that filled out the survey. So we're trying to get more responses in, and we'll probably maybe bug you by email over the next week or so. Renee, I'll pass it to you for one of your updates.
Thanks, Elliot. I just wanted to make sure everyone was aware we're hosting a special networking event on November 8th, designed specifically for scientists and the alternative protein research community. This is for scientists and academia, industry, students, really anyone with an interest in alternative protein science. I'm going to drop the registration links here in the chat. We're hosting two different sessions, one at 11 a.m. Eastern and one at 8 p.m. Eastern, both on November 8th. So hopefully one of those times works with your schedule. The event will start with breakout rooms. So there'll be a cultivated meat focused breakout room so you can connect with other people interested in cultivated meat science. And then the second half of the event is one-on-one networking. And you can choose, like if you meet someone in that session room you'd like to talk with further, you can do that on the platform. So it's kind of a good way to grow your network. And we hope you'll be able to join us, and do let me know if you have any questions. Thanks.
Awesome. Well, for today's speaker, I'm pleased to welcome Sarah Bolmer from Triplebar Bio, who's going to be talking today about some of their cell line engineering platform work. So feel free to drop questions in the chat throughout the presentation, and we'll have time at the end. So Sarah, I'll turn it over to you.
Awesome. Thanks, Elliot. Hi, everyone. My name is Sarah Bolmer, and I'm the product lead for food nutrition at Triplebar. Before I jump in, I just want to say a thank you to Renee and Elliot, and frankly, all of GFI. I think you guys have put together such an amazing network of information and kind of pulling all of this together to kind of make it widely available to the industry, which is just so wonderful. I think the other thing that you guys have done is build a network of folks. And I have to say, I think this industry, particularly in food, I have loved the network of people that are trying to bring science and new technology to solve these problems. So thank you to you guys, and thanks for having us today to talk. So to give just a brief intro on me, my background is in chemical engineering, and I've spent my career in industrial biotech and precision fermentation. And I was at DuPont working on industrial enzymes, doing kind of discovery, development, as well as manufacturing for a number of applications. I also spent a good bit of time at a company called Zymergen, which is a synthetic biology company working on high throughput engineering to make bio-based products from precision fermentation. So in my career, I've gotten to spend a lot of time and really kind of, have the bug for the bioeconomy, and have just kind of fell in love early with the idea of using biology to be able to make products that are better for people as well as the planet. But I think while I've been able to be a part of a few of the success stories of the bioeconomy, I think more than anything, what really kind of lingers in my mind is kind of the amount of headache and sometimes heartache of trying to use biology as a product development tool. And so one of the reasons I joined Triplebar is what we're really focused on is building a platform that enables us to look at much more of the landscape of what biology has to offer. And the real focus there comes to say, how can we actually take the massive search space that a genome of a microbe or an animal cell gives us and use that to our advantage, as opposed to it being this complexity that is too hard to untangle. So we will jump in and talk a little bit more about, particularly we're using this in the cultivated meat space. So yeah, I'll jump in now and do a presentation. Elliot gave me the heads up. I'm the only speaker today, so hopefully it's only take about 20 minutes or so, and I'd love to field questions and get the discussion going. So please feel free. And I haven't met most of you on this call, so it'd be great to meet new faces and get the conversation going. Okay, I'll pop the slideshow. Just to check, good on slides?
Looks good.
Yep.
Fantastic. Okay, so what I'll be diving into today is really talking about how we're using our platform to drive what we call hyperspeed evolution to unlock the suspension cell line phenotype for cultivated meat. So we're really focused on this phenotype as kind of at the outset, this kind of big challenge of taking adherent animal cell lines and taking them to suspension, preferably single cell suspension, so they can grow at the costs and scales that will enable us to really bring cultivated meat and seafood to the masses at a cost that makes sense. Wonderful. So before I jump into the specifics on cultivated meat, just a little bit of a background on Triplebar. Really, our mission is working on products that nourish, heal, and sustain people and planet. As I mentioned, we're a platform company that's really focused on how to build a system that allows us to look at much more biological diversity. And the way we do that is by really scaling down kind of the unit operation of the lab. You can think about that as a test tube, and allowing us to do way more screening, really look at millions and millions of biological systems in a day. And so that kind of system gives us the power to kind of actually apply this to a lot of different biological systems. So we have the opportunity to work on quite a number of things that kind of cross both food and the pharma space. So I'll talk much more about what we're working on in cultivated meat. The focus there really is on the development of cell lines that will enable us to hit the cost and scale that will make this impactful. We're also focused in food and bioactive proteins. Our work there is really driven around how can we optimize the microbial systems that are used in precision fermentation to make some of these proteins that are known to have really amazing health outcomes, but we need to be able to make them at costs and volumes that will allow them to make their way into more products and then ultimately reach more people. And then actually the third thing we're working on is in the pharma space and is on antibody or biologics discovery. And so using our platform to engineer and discover biologics that will modulate cell behavior specifically to improve disease outcomes with fewer side effects. So that's kind of who we are as a company. We often get asked how we get to work on so many diverse things. We often ask, this seems like, how could you do all this and still stay focused? We really like to think of ourselves as one of the more hyper-focused companies in this space, because what we really focus on is where we are in the value chain. And I think Elliot mentioned this with the different kind of groups and how we're building this ecosystem, particularly in cultivated meats. We really sit on kind of the front end of that ecosystem. So we are focused really on the development of cell lines, of the biological systems that are going to make these products a reality. And because of that, we are really focused also on working with the right partners who want to make those cell lines into a food product and manufacture and actually make them a reality, to show up on people's shelves and in their kitchens. So we're really focused on that kind of upstream part of this value chain, and are frankly really excited to see kind of this development of the cultivated meat ecosystem, and more companies start to think about where they really want to play in the specific parts of the value chain, and how we can bring kind of groups and coalitions together to make these products really successful. Okay. So to tell you a little bit about some of the team that's working on making this a reality in cultivated meat at our company. So as I said, I'm the products lead for the food and nutrition at Triplebar. I also hope you get to meet some of these folks in the GFI community going forward. They're great and are really kind of driving a lot of the thinking that we're doing around cultivated meat at Triplebar. So we also have Cynthia. She's our project lead for this work. We have Jenny Tung. She's been at places like Finless and Upside, one of our staff scientists in this group. Bin Zhang has been in kind of high throughput screening for a very long time, places like BioRad. And Kosuke is one of the main microfluidics scientists that we have at Triplebar, and has been really adapting our microfluidics technology to work for animal cell lines, and how we adapt it to the kind of phenotypes for animal cell lines that we want to go after. Okay, so really, that's our team. So what are we working on at Triplebar in the cultivated meat space? So having been in this space for a bit, I often get to have conversations with folks about what it will take to actually be able to make cultivated meat work at scale. And given that so much of this is happening for the first time, I think the challenges often seem many. You know, we hear things like media costs or bioreactor volumes, ability to actually scale these cell lines to work anything beyond a pilot. Or that we need to use microcarriers in fermentations. And so I think what I often think about is that so many of those problems really come back to ultimately is the same key bottleneck. And that's ultimately that animal cell behavior has not been, not been evolved to the system we're trying to use it in. And if we think about that, it makes, kind of complete sense. Animal cells have spent millennia evolving to work in the environment that they are in now, which is, to grow in an animal, often grazing in a field or swimming in an ocean. And they have not been optimized to grow in a fermenter at really high viable cell volumes and fast growth rates and with low cost media. So thought about that way, it's not, a crazy outcome that those cell lines that are not, or that are evolved for a completely different environment don't work that well in the environment that we're asking them to work in, which seems like could be quite a Herculean task to get those animal cells to grow in that new way. So at Triplebar, when we're faced with a difficult problem, we turn to kind of the solution that biology or nature has used for millennia, and that's evolution. I think evolution has solved so many problems and does so without requiring a mechanism. And we kind of only need to look around at the world to see this. And I am repeatedly kind of reminded of examples of things that evolution has reached kind of amazing jumps and leaps and bounds. Our founder and CSO, Jeremy, always liked to talk about that birds have actually evolved ways to almost see magnetic fields and use them for navigation. I think, even it's not so far to go to even human consciousness has kind of gotten to where it is through evolution. So in the grand scheme of things, taking a cell line from adherence to suspension doesn't seem like such a big jump, looked at with that lens. The only problem with evolution, and I've worked it up quite a bit, is that it takes so much time. And with the pressures of speed that we're trying to get to in the cultivated meat space, the time it would take to really let evolution find its way to the cell lines that we need is just too long to really make the impact that we need to have. We need to have the impact now. We needed to have it, yesterday. So we really don't have time on our side. Naturally, many companies and many talented scientists are turning to the tools that we've used in the synthetic biology industry and the pharma industry, things like genetic editing or viral immortalization or CRISPR, to be able to kind of shortcut evolution and say, how can we get to these outcomes faster? How can we give ourselves a better chance of success of hitting these? At Triplebar, we don't think we have to use those tools. I think, our belief is that, we have more than enough hurdles to overcome to get the broad consumer acceptance that we want to have impact with cultivated meat. And to add GMO on top of it is just one more thing to kind of add to that set of hurdles. And so our belief is if we don't have to use genetic editing, why would we? Why not just be able to use evolution? But if you want to be able to do that, you need to have a system that allows you to use evolution on a time scale that can have product impacts. And so we've done quite a bit of thinking about that at Triplebar, unsurprisingly. And really where we would kind of come back to is ultimately the ideal system that would allow you to make evolution work for cultivated meat would enable you to be able to screen vast amounts of random diversity and do that quickly and reliably. And for us, that really we think about kind of four main characteristics that screening platform would need to have. First and foremost, it needs to be able to screen an enormous amount of diversity. And in our mind, we often think about that as being able to oversample the animal cell genome. For us, that's critical because we're asking for kind of big phenotypic jumps in biology. And so to be able to get that, we need to be able to make sure that the solution is in the library or the solution space that we're looking at. And ultimately, to ensure that, you need to not just, have 10% or 5% of the genome evaluated. You ultimately want to be able to oversample all of the options you have in the animal cell genome. So really that means that you need the ability to screen on the order of billions of mutations and do that at a feasible cost and time scale. We also often think about wanting to be able to evaluate individual cell lines as opposed to populations. And that gets us to a place where we can evaluate cells on a kind of clonal cell level and quickly isolate towards the cell line that's going to have the impacts we want it to, and not just a population of tens of millions of cells that are in a kind of single population. We also want to be able to quickly identify improved cells. So really, ultimately, you'll want to see a cell line improvement or an outcome you care about in just a couple of doublings, as opposed to having to wait tens or hundreds of doublings, which for animal cell that can have a doubling time of 48 hours can be months of waiting without a guarantee that you have a success at the end of your screen. And then finally, we really think about this last one about maintaining a diverse set of solutions to be able to achieve a fitness maximum much faster. So to get these animal cell lines to work at scale, we're going to need to ultimately stack many different traits on top of each other to reach kind of cost and scale. It's not just going to be suspension, unfortunately, or not just going to be a fast doubling time or, ability to grow in low cost media. And so to be able to ensure we can stack those number of traits, being able to keep a lineages open and keep the number of winners open so that you can actually climb the fitness landscape across many different phenotypes is critical. So as we think about these four attributes, kind of like how do our typical how do our typical tools stack up? So unfortunately, they kind of have a number of limitations, which is why Triplebar is excited to bring our technology to the space. So, I mean, if you take shake flask evolution, I think one of the amazing things about shake flask evolution is you can test a large amount of diversity. You can put many millions of cells into a shake flask. The problem is that you have to evaluate those as a population. So you're not looking at individual cell lines. You also require often many, many doublings to see the shift in an entire population towards the phenotype you care about. And that takes a lot of time. And then even at the end, if you're successful in your screen, you're often left with, just a single lineage coming out of a shake flask, or very low numbers to keep your kind of lineage tree open to many different phenotypes. On the other hand, we have liquid handling robotics, and they solve some of the problems of shake flask evolution. In particular, you can look at, individual like clonal populations. And it's a smaller volume, so you don't need quite as many doublings to see a shift in your cell line or a phenotype. But the drawback with liquid handling robotics is, while it's faster than just hand pipetting, we're still not at the scale to be able to screen billions of random variations. To be able to do that, it would honestly take far too long and too high of a cost. So, naturally, many folks that are using liquid handling robotics for engineering are turning to things like genetic editing to increase their hit rate so that it doesn't take as much time and cost. But as we said, I think one of our main focuses is how can we get to the cell line outcomes we care about without having to use genetic editing tools and just use kind of random variation. So that brings us to our platform and really what the crux of the screening we're trying to do is. So we have built a platform that aims to ultimately run evolution at hyperspeed. And so we often liken this to kind of the microprocessor chip that we use in computation. So it used to take, an entire room full of vacuum tubes to be able to process the information that, we can now process in our smartphones. And what enabled that was the miniaturization down to the microprocessor chip. We often think about kind of the bioeconomy in a similar way. Right now, some of the state of the art is screening biological systems and with robots that often take up the size of an entire room. Our platform at its core is miniaturizing that so that we can do the level of screening that would take an entire room of robotics and do that in a microfluidics chip, in the chip that, you can fit into the palm of your hand. And so we really kind of liken this to being kind of a microprocessor for biology and enable us to screen much larger, vast sets of biology than we are able to do with kind of more traditional approaches. So that's the kind of core of our technology, but what does it look like applied to animal cell lines? So what we've been working on is kind of adapting our tech to be able to work with animal cell lines. And most recently, I've been really focused on the suspension phenotype as kind of a jumping off point to say, can we affect some of the key criteria that we need to improve with animal cells to get them to scale? So what does this look like in our process? So really, unsurprisingly, it looks like a kind of build, test, analyze, and repeat cycle, much like evolution does naturally. And really that first step for us starts with using random variation. And we generate that random variation at the scale and magnitude of what our screening capacity can handle, which in our case is kind of billions of variants. So you can think about, we get to look in kind of a single generation on the order of 1 million genomes. Those genomes can hold, hundreds to thousands of variations per genome, which takes us to the order of billions of variants. So we're already at the kind of magnitude of an animal cell genome, which can be in the range of billions of base pairs. We can then, we take that diversity, all of this random diversity, and put this through our system where we've built assays to screen for the phenotype or the output we care about. So in the case of suspension, we're able to see in our system cells that are growing in suspension versus not growing, or not growing in the way we want them to be growing. If they're growing in aggregates, or can't grow in single cell suspension. And so we've developed the ability to screen that phenotype on the range of hundreds per second, which allows us in a single generation to look at something like 3 billion variations. In that process, what we can do is quickly sort out the things that have the phenotype we care about from the things that don't. And so you can see from the picture there, characterizing things as either in the keep bucket, which would be things that you are getting to suspension, from things that are not have the phenotype that we care about. And so through that selection process, we then enrich in the phenotype we care about, in this case, suspension. And that allows us to then take those winners and analyze and characterize them, which kind of infuse us with new learnings and new understandings around those cell lines and what enables cell lines to get to the phenotypes we care about. But it also allows us to kind of go back to the beginning of the process and really start to run these iterations, much like you would with an evolutionary process. And so we can take this back and continue to evolve and improve the phenotype we cared about. Or we can start to stack new traits and look at things that will enable the cost points in different ways. Things like improving doubling time, improving the kind of max cell densities, or even things like growing in different low-cost medias or specified process conditions. So that's what our process looks like. I'm happy to say it's more than just theory. We've actually been successful in taking cell lines to suspension. Using this platform in the process I just showed you, we've taken a mammalian cell line from adherent to suspension growth. In a single generation, the numbers I kind of quoted there above were actually from a case study. We screened the effect of roughly 3 billion mutations, all randomly generated. And in that single generation, we were able to discover cell lines that display the suspension trait. So, the proof is in the pudding here, and we're really excited about this outcome. And we are excited about starting with something difficult like getting from adherence to suspension. But one of the things we ultimately wanted to test with the system was not just can it necessarily find these phenotypes, but is it able to find really rare phenotypes? And particularly with the difficult things we're asking these cell lines to do, we expect that the actual outcome we want to be quite rare. So one of the tests that we put together was to see if our assays, if our process can actually find really rare events by directly detecting the desired outcome we were looking for. So one of the things we did was to actually build kind of a mock library, a test library that we wanted to evaluate our ability on. So what we did was put this mock library together, started with one suspension cell line to a ratio of 10,000 adherent cell lines. And then we kind of directly looked for that phenotype, as I mentioned before, at very high throughput. And what we wanted to test for was our ability to not only find that rare suspension event, but ultimately enrich a library in that phenotype compared to kind of more traditional methodologies. So what you can see on the right here is a little bit of data from that experiment that we can show on this presentation. So what we were able to do with our process was enrich the population in suspension by more than 1,000-fold. And that means that we end up with a population that is highly enriched in the phenotype we care about. It means that we had many winners in that population as well. And so you can see we also compared that to kind of the no Triplebar process or more traditional, more traditional methodologies. And, pleasantly, we see almost a 2 folds of magnitude difference between our ability to enrich in a key phenotype versus not having that process in hand. So we found this really encouraging, and I think we are really excited that we use suspension here as a case study. But really, ultimately, we've been developing this platform to work as broadly on kind of the phenotype outcomes we care about in animal cell lines. And so we really think about, the full set of commercialization outcomes we'll need to make these cell lines work at scale and at cost. And so we can think about, adapting all of our screens to those key phenotypes. So we often think about things like shortened doubling time. This is a thing that evolution, notoriously very good at finding things that grow faster. Our ability to kind of look at ability to measure doublings at a set time point and enrich for things that have faster and faster doublings on a much grander scale allows us to kind of make rapid improvement in things like doubling time. We also think a lot about low-cost media. It's a challenge that comes up so frequently. And, how are we going to get the costs of growth factors down? How are we going to get the costs of, all of the expensive medias that are typically used for animal cells? We often like to think about flipping that a little bit on its head. How do we actually evolve the cell line to work in more low-cost media or in more stringent conditions without those expensive components, or with as little of those expensive components as possible? And actually think about that as an evolution problem of evolving these cell lines to work with less and less of those expensive components, or in the specific growth conditions you care about. Obviously, high cell density is really important. Getting as much cell mass out of a bioreactor as you can is kind of the name of the game. So questions like, how do we increase the cell line's ability to work in the presence of waste products and grow in the presence of waste products, or even produce less waste product so they can grow to higher cell densities? Can also kind of directly screen for cell lines that make it to very high cell densities as well. And then there's, other scale-up considerations. And frankly, I love having these conversations with people, because every time I do, there's other problems that come up that we get to think about and see if we can find ways to assay and improve cell lines towards those other scale considerations. As someone who worked in fermentation for a very long time, I often think about heterogeneity of these large bioreactors. What cells are seeing in one part of your large bioreactor will be different than another one. So how can we find cell lines that are robust across a variety of pHs or temperatures or oxygen limitations? So finding cell lines that are evolved to work not just at your perfect condition, but actually at a range of conditions so that they're robust to excursions or to the differences you see in a large bioreactor. So those are kind of just some of the things we are thinking about doing. And what I'll end on before I go to questions is kind of one of the things that I always come back to with this platform is kind of the magnitude of diversity that we can look at when you look at a system like ours, as opposed to something like high throughput screening with robotics. I spent a lot of my career doing high throughput screening with robotics, and it has certainly been a step change over kind of manual pipetting and manual liquid handling. But unfortunately, and I've lived this, unfortunately, it is still not scaled to the set of diversity we have in our hands with biology. And so we think about the real improvements we'll make or when we can truly oversample an animal cell line genome. And oversampling means getting in the range of 1 to 10 billion variations. And so we often think about that as screening in the range of at least 10 million cell lines. On a system like ours, we can do that in just under 2 months with generations of 1 to 2 million cells and stacking those on top of each other. If we compare that to what it would take to do in, even something like a 96-well plate, to get to 10 million cell lines, you're screening more than 100,000 plates. And to put even aside just the cost, the sheer magnitude of cost of that, the time alone would mean that even at very good throughput and operations of 100 plates processed per day, which is what you're doing at some of the best companies that are doing this, it would still require 4 years of screening to do an entire genome. And so that's just not, frankly, viable in a world in which we're trying to get speed to get these cell lines and get them to market and into the consumer's hands and have the impacts we want to have. So that is all I have. And so thank you for taking the time to listen today. My email is there. I would love to connect and talk more. And yeah, I'm happy to take questions.
Awesome. Well, thanks, Sarah, for that presentation. And hopefully some folks will reach out to you to continue some more discussions online. Super interesting technology that could be really useful to a lot of people. So thanks for sharing.
Awesome. Thank you for having me. Bye, everyone.